Single-molecule FRET experiments using surface-bound probes have been performed almost exclusively on immobilized molecules in the past. However, many biomolecules diffuse and can be analyzed with our method.
Combining single-molecule FRET with tracking makes it possible to analyze not only FRET efficiency time traces of moving probes, but also to investigate spatiotemporal aspects such as the diffusional behavior.
Our method is compatible with a variety of different FRET-based probes. For example, it could provide insights concerning molecular forces, conformational dynamics, and binding kinetics in live cell experiments.
High-quality single-molecule FRET experiments are notoriously hard to perform. For reliable quantification, it is important to record data at a good signal-to-noise ratio and single-molecule tracks of sufficient length.
To begin sample measurement, excite the donor and acceptor fluorophores for an appropriate illumination time while triggering the camera and wait until the camera readout is enabled. Repeat the excitation of the donor and acceptor fluorophores alternatively. Choose the number of repeats to be large enough to ensure photobleaching of at least one fluorophore per probe within the field of view, permitting stepwise photobleaching analysis to discriminate single-molecule signals from aggregates. Specify the illumination sequence to permit the selection of donor and acceptor excitation frames, as well as frames for image segmentation from recorded image sequences. Simultaneously analyze the datasets recorded using the same illumination settings. To this end, assign